available online to follow up when we post all this stuff up on the NBCEC website. Our 2018 session runs for 4 weeks here in October. If you go to get the recorded sessions, those will be posted under the For Professionals tab and Brown Bagger link on the NBCEC website. So be sure and visit that. The cloud recordings once in a while take a little while to get processed, but we'll get those posted up as quick as we can if you want to share that with colleagues. This week, I'm hosting This Week and Next. I'm at K-State and been an active participant in helping coordinate the Brown Bagger series for quite a while, and again, we're certainly glad you all joined us for today's program. Today's session focuses on recent changes to genetic evaluations. We'll have actually kind of two connected talks by Dr. Bruce Golden of Theta Solutions, and those two talks center around single-step implementation using marker effects models, and then a second portion focused in on stayability and sustained cow fertility. By way of introduction, Bruce is a co-founder, CEO, and president of Theta Solutions. And prior to founding Theta Solutions, Bruce was the chief executive officer of a biomedical instrument technology company that he founded after a 19-year career on the faculty at Colorado State University. That's where I first got to know Bruce when I was an undergrad. While at CSU, Bruce was a full professor in animal breeding and genetics. Dr. Golden has also been on the faculty as the department head at Cal Poly San Luis Obispo, where he innovated novel graduate degree programs and raised substantial fundings through research and advancement. Dr. Golden's research and service to industry has resulted in numerous awards and publications, including a series of patents related to animal identification and diagnostic instruments. While at Colorado State, he was the founder of the Center for Genetic Evaluation of Livestock, or CGEL as we called it, and they're a provider of genetic evaluations for breed organizations and breeding companies worldwide. Bruce has extensive international experience in livestock production, including work in China, Brazil, Venezuela, New Zealand, the UK, Canada, and Australia. Dr. Golden attended Washington State University in Pullman and received both a Bachelor's of Science and Master's degrees in animal science. And he received his doctoral training and degree in animal breeding and genetics from Colorado State University in Fort Collins. So with that, Bruce, the floor is yours. I'll unshare here. We appreciate you joining us today and look forward to your comments.
Great. Thanks, Bob. Here, let me get my slideshow up here. Get it started. Thanks for inviting me. I really enjoy these. I'm really impressed. The 14 years of this program. That is sustainability right there.
Right.
Yeah, stayability. That's excellent. Great. Well, I'll go ahead and get started. I promised Scott Newman that I would keep my dogs from barking in the background, so you'll excuse me if I'm not successful at that. I'm broadcasting from home, and hopefully my mouse pointer is moving fluidly enough, and the technology holds together long enough here. I want to start by first talking about who we are a little more. Our team consists of myself and Dr. Dorian Garrick, who most of you know. And we've recently, about a year and a half ago, we brought in Daniel Garrick, Dorian's son, who received his PhD in aerospace engineering. Very smart guy, very, very good software developer, very good with customers. And, you know, Dorian and I are getting a little long in the tooth, and it won't be sometime in the next 10 years, we'll hand off the whole thing to Daniel and his partners, whoever he chooses at that time. But we're not ready to leave yet. The company, Theta Solutions, not only offers software licensing of our Bolt software, but we also offer support and model development and training. And a big new area that Daniel has led is the curation of genomic data. And we have a number of customers around the world, not just in animal breeding, who we curate genomic data for. All right. So as Bob mentioned, we're gonna talk about our implementation of the single-step marker effects model. And I'm going to focus on 2 of our roughly 15 customers. I'm going to focus on the IGS team, which is the large international genetic evaluation headed up by the American Simmental Association. But I'm also going to talk about our work with the American Hereford Association. That's a really fun project and allows us to do a lot of additional research. And I'm going to show you a little research that we've been working on their data. Along with this implementation, I want to talk about the effects of our new calculation of accuracy and what that does. I just need everyone to understand what we're doing there and some of the things that you will be seeing or have seen in the accuracy calculation. And then I'll wind it up. We'll take some brief questions at that point, and then I'll wind it up with just a few minutes on the new approach that we've got to stayability and sustained cow fertility. All right, I always like to start these talks with a discussion of what the enabling technologies are that allowed this next generation of genetic prediction to occur. You know, we've really ratcheted up our game by implementing technologies that were actually originally built for the computer gaming industries, and a lot of scientists in the last 10 or dozen years have figured out that these high-performance graphics processing units and high-performance processors and motherboards and other components really allow us to do a lot of computation in a relatively inexpensive platform. And so that's kind of one driving technology, but obviously the big driving technology that we're mostly focused on here is now the proliferation of relatively inexpensive, and that's relatively inexpensive SNP marker technology. Think of the bead chips that everyone is using, and I'll show you some numbers of how many we're up to here in how many genotyped animals we're up to in various analyses. But by way of review, it's always good to review this. I know most of you know this already, but the cattle genome has about 3 billion base pairs in it that represent about 22,000 genes ostensibly. 98% of the genome is non-coding regions in cattle DNA. There's about 6 million SNPs more getting discovered all the time. And a SNP is just a single nucleotide pair that is different from animal to animal or chromosome to chromosome at the same location. And those are used as markers, and the vast majority of the SNPs that we're using now in these single-step evaluations are actually in the non-coding region, and the ones where we get good signal are near, approximately near genes that have an effect on the traits of interest here. Now, I'm going to show you some things about that and talk to you a little bit about that. I want to use this opportunity to plant some seeds. I know many of you will be at the Genetic Prediction Workshop in Kansas City in December, and we're going to talk in more detail about some of these ideas. But I'm going to show you a little bit about how we think this is actually working among these SNPs. All right, so we've implemented, so those are the enabling technologies that are kind of mechanical here, right? High performance computing, that always gets better and always allows us to do more and more complex things. And then all the technology behind getting DNA markers, SNP markers. But there's been some other innovations. There's a couple of different, several different ways to skin this cat to implement a single-step evaluation. The way that we have implemented it is based on some work Rohan, Fernando, and Dorian, and Jack Deckers, and I was involved in parts of that, and a few other people did, in developing what we call the single-step super hybrid model. And if if you're familiar with our papers in December of 2016 in GSE, we published a paper that showed two different formulations, equivalent formulations of that same model. And I'll show you the formulation that we've actually implemented. It's the two formulations just differ only in what's the more sensible approach to take given how many genotyped animals and non-genotyped animals you have in the analysis. And as I said, I'm going to focus in this talk on the work with IGS and AHA. And you can see the numbers there. We've got nearly 13 million animals as of August 21 in the evaluation, and about 140,000 of them are genotyped. And that's in August. These evaluations are run weekly for both Hereford Association and IGS. And so these numbers are coming up pretty quickly. You can see as of last week's evaluation in the Hereford Association, we're focused on 2.3 million animals with nearly 50,000 of them genotyped. And these numbers come up pretty quickly, especially this time of year. A lot of data is coming in now into these programs. We implement these evaluations as multi-trait evaluations with multiple components. So think weaning weight directed maternal, for example, the usual thing. But then we also implement as a component an extra polygenic effect. And I'll talk quite a bit about that. That turns out to be actually the extra polygenic effect, or EPE, in these analyses actually turns out to be a pretty important component because of the way we're seeing these SNP panels work. And I think that'll become a little more clear to you in a few minutes. All right, so for those of you who are disinterested, now's the time to take a break and pour another cup of coffee. I've only got one slide of models and equations, but I was assured that it would be a good idea to show it for those who are interested, because there are a couple of ways to implement this model, and I want to show you our specific way. So if you formulate the model this way, and this should look familiar, this is straight out of our 2016 GSE paper. This is a single trait implementation where N means non-genotyped animals and G means genotyped. And over there on the left, then you can order things this way. You don't have to. The actual implementation is done a little differently, but order it as observations on non-genotyped and genotyped animals, and then X is the usual incidence matrix relating fixed effects in beta to the observations in Y for non-genotyped and genotyped. That Z U sub 1 minus C, well, that's the extra polygenic effect here. That's the effect. U sub 1 minus C is the effect not accounted for by the markers. So the additive genetic effect not accounted for by the markers. And it turns out that's a pretty important effect, because in our current marker sets, our current DNA chips that we have access to, we're really getting, and I think you'll see this, not much more than 50% at maximum of the amount of genetic variation described by the markers. And that's relatively well-known kind of effect. C here represents then a partitioning coefficient, and it's basically how much a priori do you assign to the extra polygenic effect and you assign to the marker effect. Now, it turns out these models are pretty insensitive to what you assign C to, especially with these large numbers. The data pretty much, it acts as a prior, and the data pretty much drives that partitioning to where it's appropriate. So then we have Z, and I put a little star by the incidence matrix of relating, in the case of ZN, the animal effects. So think of U sub C as the breeding value effect on non-genotyped animals due to the genomic marker effects. And there's we use imputation, and the formulation of the model allows to predict a genomic effect for non-genotyped animals through their relationship to genotyped animals. So ZG is the incidence matrix relating animals who are genotyped to observations in YG, but it does it through MG, which is the genomic matrix, right, where the columns are the markers, and the rows then are the genotypes of each animal who has a genotype. So that alpha then are the marker effects, and error E is the usual residual effect. So if you look at the formulation for the mixed model equations on that, you can see if, I'll try and use the pointer here, I'm not sure you're seeing that, but I'm over here at the second diagonal block, and that should look really familiar to those of you who are analysts listening here. That is really just a blup, right? And so that's for the extra polygenic effect, U hat sub 1 minus C, where C is this partitioning coefficient. And again, this is a single trait case. It's, you know, obviously a little different when you're in multiple trait case situation, but you can see that that's the lambda ratio, that's the usual inverse numerator relationship matrix, and the lambda ratio, the ratio of sigma squared E over sigma squared G, but in this case, you partition the sigma squared G by the proportion, the a priori proportion of, oops, I got it, C minus 1. That's supposed to be 1 minus C there in that slide. And so really that's just a blup on the, and that's the usual blup for the genetic effects that the markers cannot account for. Okay, then you move down to the next diagonal, that's the third diagonal down, and that is then the diagonal element corresponding to the U hat sub n. That's the non-genotyped animals' proportion of their breeding value that is accounted for by the marker effects, and that's done through, you know, this imputation step. And I think once you look at Q, the total diagonal of the alphas, that's in the lower right corner of the left-hand side matrix, I think you'll see the imputation, or at least implied the imputation there, because Q looks like this. Q is it looks relatively complex, but it's not that complex. You can see it's a function of 3 pieces here added together. The first piece after the equal sign on the left side of the right-hand side of the equal sign is just the mg prime mg bits, but notice that it's multiplied in the middle by the incidence matrix z sub g prime z sub g. And what that becomes is, in these additive direct effects, is a diagonal matrix of basically zeros and ones on the diagonal. If it's a conventional model, you know, if it's a random regression, it becomes something else, you know, those sorts of things. But think of it in the simplest case where this is a conventional model, has become zeros and ones. And what that has the effect of doing, and this is very important, it picks out and uses only the genotypes on animals with observations in Y. And so it's that, especially in multi-trait cases, think of that, think of it as not every animal is going to have an observation on every trait in Y. And that's an interesting feature of this model, that it uses the genomic data. Of course, it's sensible only when an animal has an observation to pick that out. This is just a diagonal element where sigma squared alpha is the variance genetic variance due to the marker effects, and you know, you can think of that in a couple different models. I'll talk a little bit about how we actually implement that and what sigma squared alpha is in our situation. And then over on the right hand side is where mn then is the imputed values from relationship with mg for non-genotyped animals. And that looks hairy. You know, in our case, we have, you know, 12 million of those kind of animals and 150,000 in MG. And MG is a relatively small matrix compared to MN. And so that looks like it's going to be huge. Now recognize that Q is number of markers by number of markers, and that's its maximum. No matter how many genotyped animals you ever have, Q is always going to be the same size. It's just going to have different numbers because of how many genotyped animals you get. So MN looks pretty hairy here, but the reality is through some pretty interesting little algebra that Rohan and Dorian worked up, you can calculate. You don't actually ever have to calculate MN. You can do MN ANN through your imputation step, and that then makes because MN is equal, MN prime ANN MN is equal to that thing on the left, the negative MGAGN MN. That comes out to be, you calculate the left-hand side of that AG and MN first during the imputation step, and that's actually a relatively small matrix. That's number of animals genotyped by the number of markers in the model. So it's genotyped animals, not genotyped, and then you just multiply it by MG. So you can see this model, this implementation, this version of the model is really efficient for situations where you have a big difference in the number of genotyped versus non-genotyped animals. If the number of genotyped animals is relatively small, this is a really efficient implementation and is very easy to put together and fast to put together. Okay, so that's the math. Just wanted that in there for posterity for everyone so you knew which version we were using, because as I say, there are actually several. So out of this model, the EBVs are calculated as the genomic effect plus the extra polygenic effect. So for genotyped animals, that is M alpha, so their MG alpha, plus their EPE for genotyped animals, the extra polygenic effect. And for non-genotyped animals, you solve and you calculate their EBV as UN, U hat in, plus their extra polygenic effect. And of course, had to put this on the bottom. EPDs are 1/2 the EBV, no matter what. There are a couple other things that go into this equation. That's the most simplified form, and I'll talk about things like the J and the K equations, which are in our literature. Well, I want to talk real briefly. This is where I want to plant the seed for the GPW. A lot of people have questioned our use of not using full marker sets in genetic evaluation. We use subsets of markers in actual production of genetic evaluation. I want to try and convince you or get you to start thinking about why that is actually a better method than trying to fit all markers. So let's say we get panels of all different densities up to very high density and very low density, and we're imputing right now up to one of the 50K panels. It depends upon which evaluation you're doing, but one of the versions of the 50K panel. And out of that, then once we impute to that, we pull out subsets of markers to fit. And here's kind of the reason why. If you look at the top plot, the alternating colors starting on the left are the first chromosome is red, second chromosome blue, red, blue, out to the 29 autosomes in this marker set. And this is all 50K in this plot, all 50,000 markers. Fit, and the vertical axis is just the absolute value of the alpha effects. This happened to be on Hereford birth weight and on a centering project that I've been working on that I'll also present at the GPW. But you can see, if you go to the bottom plot, that's the posterior probability of inclusion or posterior inclusion probability for those markers. And so again, it's the chromosomes, and then the dots align with the dots in the upper plot, whether that's the marker effect. What I want you to get out of this, what the take home is there are some very large QTLs on birth weight, and those are all well known and their positions have been well documented for a long time. But then the vast majority of markers, by far, the vast majority of markers have no detectable effect in this evaluation. They're virtually zero. It's really only a relatively small handful of markers that actually are associated with this trait, in this particular case, birth weight. And you can see that by looking at all the markers that are way down at the bottom of the plot, the vast majority, tens of thousands, well, nearly 50,000 markers have no discernible association. And if you look at the PIPs for these markers, you can confirm that the probability of including those markers in this set is very low. And we also confirm this through doing, those of you who are familiar with window variance calculations, I won't show them here, I'll show them at the GPW, but through window variance computations that allow us to verify the fact that only very few markers are actually, relatively few markers are actually having an association. So so I take those, and in this previous plot, those were fit using a Bayes C with a pi of 0.95. This is relatively invariant to what pi you fit. And as I said before, we have a starting prior of 0.5, but really it's very insensitive to what that starting prior is. And we calculate these through what's called a variable selection or marker selection model, this Bayes C pi of 0.95. And we've played with pi. It's, again, we get the same markers out within reason of that 0.95. So this slide then shows, starting from the left, if we start including the highest PIP, and I've done it other ways, and you always end up with basically the same subset of markers. If you start including the top 1,000 highest PIPs, and actually on the far left, that was just the top 10 markers for posterior inclusion probability. The vertical axis then is the correlation, and that's the correlation between taking a training population, in this case the training population was millions of records, and for birth weight on Hereford cattle, and then getting the marker effects out of the analysis with varying numbers of markers in the subsets, and predicting the phenotypes, unadjusted or adjusted, for birth weight, and they're just adjusted for contemporary group and everything else. For birth weight in a test population, and the test population in this case was all calves born in 2017 forward, and there were I think it was several hundred, or no, it was 112,000 calves in the test population. So a nice large test population. You can argue about different ways to pull the test population, but we looked at it different ways, and it says the same thing. So what you're looking at on the far left is you take the first 10 highest PIP markers, and you're already up at a correlation of the prediction from the training data correlating at a 0.2 with the test data. And then what I did was I just said, okay, the first 1,000, you get up there and you get there in the mid 0.2 range, and then the next 2,000 you're up into the moderate to between 0.3 and 0.35 correlation. Then you grab the next 1,000 and you're staying there, and you basically stay grabbing 1,000 markers at a time. And I started jumping by 2,000 markers after the first 10,000, just it was redundant. But you can see you gain no prediction accuracy. You gain nothing in the correlation by adding additional markers in here. And in fact, we've shown that if you do a marker selection model, so you do weighted marker selection, and toward the tail, you actually get a decrease in the correlation of the markers selected to the marker to the Bayes C0 analysis of all markers in the case, and it's just a slight drop, but it makes sense. You're overfitting things, you're over parameterizing the model. So the take-home message is the marker subsets, at least with the markers that we've got available to us now, are as good, if not better. And I'd say theoretically they are slightly better to fit just subsets. Well, you can see what that does to this model, where we'll have that diagonal block of Q is only number of markers fit by number of markers fit. It turns out to be a very relatively small problem, even in large multi-trait models, and relatively easy to fit with no sacrifice in accuracy, and actually a slight gain in accuracy. And we've done this with lots of traits and looked at lots of traits. Another way to look at it is in this slide, I've got along the horizontal axis, standardized birth weight for marker posteriors. So each dot is a marker effect on standardized birth weight, and on the vertical axis is standardized post-weaning gain. And again, this is in our Hereford data. Again, it's a base C pi, where pi was 0.95. And here then, so this is a plot of the top 5% of markers that entered the model for one trait or the other or both. And there's about 2,000 markers here. You can see there are a lot of pleiotropy on birth weight. I don't think this is any big surprise. We have all these QTLs in the lower left quadrant and the upper right quadrant, the markers with the biggest effect, and they're unfavorable. Let's kind of zoom in, clear out a little noise on that. Here's that same plot with, or same data, only with just the top PIP. It entered at least 30,000 times in 40,000 samples. Again, all the pleiotropic markers show up very clearly, very strong signal. But I don't want you to ignore the markers that are on the zero axis, either vertically or horizontally. I propose that those are really the ones that allow us to make the genetic change. I don't think we're going to make genetic change by considering these markers that are pleiotropic and unfavorable here, all the big QTL. Now, those are the ones that are really giving us this accuracy value that we get out of genomic prediction, where we get a bump in accuracy. We get it mostly from these markers that are pleiotropic, large and unfavorable effects in traits like this. But we're really only going to make the genetic change from the ones that are at or near zero, as long as they aren't pleiotropic with something else not on this plot. So that kind of raises a question of how do we handle those markers? I don't have a good answer for you. A few of us have been talking and exploring ideas, but really we want to maximize our genetic progress by changing the frequency of the markers that are not pleiotropic here. So what does this mean in terms of accuracy? So here is for, this one I believe is for the IGS evaluation. Matti Saatchi put this together. He did an empirical analysis to say for each trait on a yearling bull who gets genotyped, how many progeny equivalents is that? In other words, how many progeny would a non-genotyped bull have to have in order to have the same level of accuracy? And it's different for different traits. The far left traits are calving ease and maternal calving ease, which is total maternal calving ease. Birth weight, weaning weight, yearling weight, milk. Then the next column is stayability, carcass weight, marbling, ribeye area, fat, and docility. And it's a different number for different traits because there are different heritabilities, there are different models, there are different numbers of animals genotyped. But you can see that the progeny equivalents in most cases are pretty substantial. There is a nice accuracy gain. Again, a lot of that accuracy gain is from those pleiotropic markers that we have trouble actually changing the gene frequency on. But these things do work. Another piece of evidence that it is working is Matti put this together, and he divided the data into training and test data, and ran a correlation of predicting or correlating the predictions out of the Bolt analysis with the old Cornell, the previous non-genomic analysis that was conducted at IGS for sires with their progenies' adjusted performance data. And you can see the Bolt analysis is substantially higher correlations in all cases, doing a better job of predicting future progeny performance. And we've seen that consistently in a number of other looks at it. And that's backed up, I think, by the previous slide where the correlation just comes up quick and flattens out. We do include other effects, of course, in these analyses. Everyone always asks, do you have breed year? And yeah, we have breed year cluster additive genetic groups. We have in the IGS analysis, in both analyses, Hereford and IGS. But in the IGS analysis, we have a lot of multiple breeds. In fact, all of these individual breed associations, such as Simmental and Geltby and even Limousin and Red Angus are really admixed populations, multi-breed data sets. So we also do include 2-breed type outcrossing equations for accounting for heterosis. Obviously contemporary group, and you know, we can dig down if you want on how contemporary groups are formed. We then include a couple of other equations. There's a J equation that accounts for the genetic merit of genotyped animals versus non-genotyped animals, and that actually gets added back into the EPDs. And then there's this K equation. It accounts for the average centering across all loci, but centering of loci is actually done much differently than just fitting a K equation. And the K equation really has no effect if you center everything correctly here. And you can read more about those in our literature if you're interested in what those equations are. So this is the list of traits that we're producing in IGS in Hereford. It's a couple of other traits are being produced instead of, we have a teat and udder trait in Hereford that's being produced. And we have a number of traits that are under development right now for IGS. Hereford already has a dry matter intake EPD. We're working on that for IGS, working on heifer pregnancy, and we're looking at days to finish, which is an interesting way to potentially account for dry matter intake. Now, with our evaluations, we do solve the mixed model equations, the model equations using a PCG solver, and they are fit as a Bayes C0 because the marker subsets, and the Bayes C0 works with the subset is equivalent to the virtually equivalent. They're 0.999 correlation on the EPDs if you do a weighted marker analysis with all markers. But we do sampling then of those mixed model equations in order to calculate the accuracy. And we do sampling because it's a direct method to estimate. It's not an approximation method. It takes a lot of work, but again, we use this hardware where the GPU hardware and high performance motherboards and processors to do the sampling. And we've gotten huge speed ups on some of our algorithms for sampling to be able to make it feasible. Having said that, it still takes about 2.5 days start to finish to do the analysis, and 80 to 85% of that time is done by calculating the prediction error variance through sampling. But it's a much higher quality estimate of the prediction error variance than what we were previously using in these approximation methods. It's virtually no bias in it. Now, that's the good news. The bad news is that means the average accuracy goes down, not up, because the approximation methods bias the accuracy up. So when you look at these evaluations, they are not less accurate, they're more accurate, and we have a more accurate accuracy. Here's an example. This comes from some of the Hereford work. This is a accuracy on the horizontal axis out of the previous Pan Am evaluation. And that was just a blip. And then the bolt marker effects model accuracy on the vertical axis. You can see the accuracy of the bolt should be higher, but the actual estimated accuracy for the Pan Am skews to the right and overinflates the accuracy. And it's especially bad for the low accuracy animals. Think yearling bulls. Traits that are multi-trait, where one trait is highly observed, such as ultrasound, and the other trait, such as carcass equivalent, is not well observed, were most impacted by this accuracy inflation. And so I think it's a real important kind of implementation or feature in this implementation. All right, I'll pause right there for a few minutes to respond to some questions. I think, Bob, are you going to field questions?
Sure, Bruce, I can field those. If you would, as attendees, the way you can post a question is use the chat box. So at the bottom of your Zoom screen here with the slides, if you just scroll over that, it should pop up a little chat button, and you can type in a question. One of the ones, and I'll start while everybody else is thinking and typing, hopefully. Bruce, one of the things I think is really interesting in these models is the use of non-genotyped animals that then get imputed genotypes. One of the things that occurred to me is, have you looked at the correlation between the UCs that get calculated for those imputed animals versus if they were actually genotyped? And I know UC is unique to the non-genotyped animals, but as we prove those animals out as genotyped versus non-genotyped, how good a job does the imputation process do in pinning those down in terms of the added contribution of the genomic information?
Yeah, good question. No, I have not specifically looked at that, but you get some gut observations. A lot of animals change pretty quickly once they get a genotype on them, and so they do a better than not doing it job, I think. But I think it's important to genotype animals, and you really get the accuracy boost. You do see, animals whose parents have been genotyped. You see they have a higher accuracy for prediction than animals whose parents haven't been genotyped. But you really get, you can get a much better accuracy boost by genotyping.
Okay, super. A question from our friend Jerry Taylor. And Jerry, I hope you're all safe with the, I just got a notification that the hurricane has made landfall in the panhandle of Florida, so hope you're okay. But Jerry asks, Bruce, when you sample markers in a multi-trait situation, do you end up with a large number of markers because different markers have high PIP for each trait?
Yeah, actually, it's funny. We haven't actually seen too much of that. So as you can see in the birth weight post-weaning gain, there's a lot of markers that are pleiotropic, a lot of markers that aren't. So there are some markers that have a higher PIP in one trait than the other. And so it is important to go through and sample each trait and peel out markers that are important for that trait and be sure that they're included in your subsets. So we don't recommend fitting, for a whole bunch of different traits, a lot of different markers. What we do is we just cluster them all together, because you can see from that correlation plot that once you get the important markers in there, adding more markers in that trait doesn't hurt you up it through reasonable limits here. It doesn't really cost you much more in compute time. But it is important to go through and look at each trait and make sure you're peeling out markers that have effect on at least one of the traits in your evaluation. I have not seen big differences in fact, I haven't seen any real differences in markers that end up in the subset, whether you do it multi-trait or single trait, but you might as well do it multi-trait. Yeah.
It's good to know.
Yeah,
great. And any other questions? And I know Matt, you're on as a panelist. If you've got one, you don't have to type it. You can just jump on with the audio component here. But others, if you've got a question, we'll certainly have some more time at the end for questions. But okay, we got one here from Milt Thomas at Colorado State. Thanks, Milt. It says genotyped yearling bull progeny equivalence for stay was 25. This approximately means that because of genotypes, the bull has an equivalent of 25 progeny records. This was surprising. Was expecting progeny equivalence to be a low value relative to the other traits presented. Can you explain or provide more information?
Yeah, that one's not super intuitive. And I wish Marty were on here. I believe that one, and I know I've asked him this, but I don't remember. I think that means 25 daughters in production. You have to have 25 daughters in production to get that level of accuracy. And so because it's a little different trait. So a couple of things about stayability. So the overall accuracy is lower for stayability. So you get a bigger bump, even though it doesn't get to the high levels of accuracy that birth weight does. So think of it this way: birth weight has a high heritability, and so it's the traits with low heritability but well-known marker effects that get the nice bump in accuracy, and that's stayability. But stayability is a weird one. I think it was Matt and I were talking yesterday about it, that or someone I was talking with. Stayability is one of the traits that's most highly impacted by markers. And Mahdi has seen that in his research too, that the markers can describe relatively large proportions of the additive genetic variance compared to a lot of other traits. And not a clear reason why on that, but yeah, that is an interesting result. It's not always intuitive, but I think if you think it through, it'll make sense to you. Next question.
Great. Another question here from Jerry, and we'll take this one and then we'll let you go on with the stay discussion because we're kind of starting to point that way a little bit. But Jerry says, what about the small effect SNPs that are hiding among the no effect SNPs? They could have low PIP because they have small effects, but could they be identified due to their having common sign effects and pleiotropy across populations and reduced unexplained EPD?
Yeah, even if those are there. So if they're a small effect SNP, but they keep entering the model, they would have a relatively strong signal on PIP. And there are small effect SNPs that do have strong signal on PIP. I have a whole set of slides where you zoom in and look at the small effect SNPs, and that you can really find a bunch with reasonable PIPs. There's a couple of ways to look for those. Window variance, we go and look. What happens a lot is an effect can get spread across a whole cluster of SNPs, and then it looks like a small effect SNP. So you've got to definitely go in and look at window variances to make sure that you haven't just washed out an effect. And there aren't many that you pick up that way, but you do pick up some. But the reality is, if it's a small effect with a low PIP, it's not going to improve the correlation, the prediction correlation. And you can see that by that plot, right? That plot of adding in additional SNPs gains nothing based on PIP, So I think that's pretty straightforward.
Cool. All right. Well, I'll stop here for questions and or stop fielding questions and let you continue. And we'll have a little bit of time at the end for a few more, Bruce, if that's all right.
Right. Yeah. Sorry, I got a little windy on that first part of the talk. I don't nearly need as much time on this part. So we've completely reimagined models for all traits, and we call it stayability in IGS. In Hereford, it's sustained cow fertility. But they're basically computed the same way. It's just these models are done through random regression approaches. The first place I saw an evaluation of random regression for stability was in a paper that Scott Spidell pointed out to me from Jem Rozic, where he did a comparison of random regression and survival analysis and a few other things. For predicting stability, and threshold model for predicting stability in using Canadian Simmental data, and that was a real eye-opener paper for us. We implemented it that way. It's obviously stability is really important, or sustained cow fertility. Sustained cow fertility is different than stability in the Herefords in that they project out to the age of 12. And it's the age of 6 in stability. That's why the names are different. But the truth is you shoot a trajectory in these random regression models. The correlation is very high between 12 and 6. So it's functionally the same kind of trait. It is the trait with the most impact in any maternal selection index it ends up in by a lot. Small changes can make a big difference. And it's because of the obvious things. You have more calves to sell, right? Higher conception rates. But then it's fewer replacement females, so even more calves to sell. And you get heavier weaning weights from the older cows. And so you get more just pounds of weaning by just having older cows in your herd, and you have less calving difficulty in it. So it really does drive these maternal indexes. The random regression that we implement uses stability on a polynomial, so intercept and a slope fit here. Cows, what I really like about this stability of random regression approach is how much better you can use the observations. And we still just use an observation of 0 if a cow did not have a calf at a given age. and a 1 if a cow had a calf at a given age, and those ages, you know, we shoot them along years, but it's fractions of years. Think of it as days. And so the trajectory is just calculated for each cow's additive genetic effects. But the really neat thing about this that wasn't a feature, well, there's a couple, but wasn't a feature in our old threshold model approaches, is it uses missing or unknown observations so much more sensibly. If you know, an animal is sold, or even if an animal enters an EG program as a donor, you call that missing. You don't have to penalize her or do anything, you know, on those kinds of censored records. It's and there are a number of situations like that where it becomes much more sensible. The other thing that's really an advantage here is in the threshold model analyses, it was a requirement. The models wouldn't behave, wouldn't converge if you didn't have, if you had to use only contemporary groups with variation in them. All cows, you couldn't use contemporary groups where all cows were open or all cows were pregnant in it. And that's a big advantage for random regression. That's not a requirement. Forcing that requirement actually adds bias. Those contemporary groups with no variation are actually informative, make informative comparisons between sires. We already said it handles missing values in more sensible ways. It's easier to implement in an animal model than the threshold model analyses are. And then it's easier to implement in a marker effects model. It's really just the same thing. You just have multiple components for each component in the polynomial. It's faster to obtain answers then. There's no Newton's method iterations. It's just a PCG solver and a Gibbs sampler as before. There's a number of papers, including Mrazek, that shows that on the same data, it actually makes better, even though MAP may be theoretically more, maximum a priori may be better than theoretically, that these random regression actually make better predictions than MAP or hazards models. And and it uses, this is a repeat measures model. It uses all the observations, at all ages through the model. Let's see, we fit in this, it's all the stuff that's in the Mrazek's paper: year, age at first calving, random contemporary groups, random permanent environment, genetic marker effects. We do genotyped and non-genotyped intercept and slope, extra polygenic effects, intercept and slope, and it also includes additive genetic groups in the model. We think you know, we've done a number of verification and validation studies on it, and we feel like it's been a real improvement in predicting stayability. With that, I'll wind this up and hand it back to you, Bob, and if there are more questions and more information anyone wants.
Great, thanks, Bruce. Appreciate that. What other questions do we have from audience and attendees for Bruce? I'll pitch one while everybody starts typing. Bruce, in these sustained fertility models, and I like the random regression approach, the far end's kind of adjust, or the predictions that you get out are adjustable, in that you could say, I want to know what the EPD is for cows at age 10, for example, versus maybe something like 12. Is that right?
Yep. Yeah, exactly. You know, we're fitting it as a 2-degree polynomial. You could argue that you fit it as a 3-degree polynomial, get a little curve going in it. But two issues there. First of all, the EPDs, when we do it, it correlates at 0.99 through those kinds of, as long as you don't get too far out on the tails, and it behaves a lot better. You start getting 3-term polynomials and you start putting marker effects in there, and then you have a covariance between each of the polynomial effects. They don't necessarily behave ideally. So we've stuck with a 2-degree polynomial. Yeah.
So the other thing that sticks out to me is, as we've started implementing these, is that the information now that we gather from rebreeding as a 2-year-old and 3-year-old on the daughters of relatively young sires compared to what we used to have in a traditional stayability model, really gets us more information a lot sooner in that bull's lifetime, so that we can make some better decisions about who we're going to keep cows out of moving forward versus, kind of the old stayability. We were, by the time a bull was proven, we were using his sons mostly as our replacement heifer generators because he was 12 or 14 years old.
Yeah, that's exactly right. There had been some methods developed at Colorado State on indexing. So you calculate, 4 different analyses at each age, 3, 4, 5, and 6, and then you index them together. But those had inherent problems too, because you had to make decisions about moving animals from one age group to another. And this just uses all that information much more sensibly in a repeat measures analysis approach. And you're right, it does give you a nice, little boost in accuracy early on, along with the genomic data giving you a nice boost in accuracy early on.
Yeah, great. Appreciate that a lot. Any other questions from our attendees? And I know this one's maybe one of the more technical sessions we'll have this season. So if you're not super comfortable in sort of the advanced animal breeding discussions, don't give up on us yet. I may ask Bruce. I knew there was going to be a lot of the technical staff from various groups and some academics on here, including, I know, our graduate students here at K-State are on. So the mathematics was to benefit those groups mostly. So if you didn't catch all that, don't worry, don't feel bad, and don't abandon us. We'll have some more applied stuff moving forward. But Bruce, I thought you did a great job explaining that stuff.
Well, I hope I didn't scare anyone off.
No, I don't.
I did ask Bob before we started. I asked Bob and Matt. Said, should I show the mixed model equations?
Yeah. What animal breeding talk's not complete without that, right?
Exactly.
Yeah, great. Well, I'll throw it out here again. Do we have any other questions from audience? We're bumping up on our end time here shortly, so if you do have one of those, throw it out. I'll Bruce, if you'll unshare, I'll throw up the slides to introduce and sort of preview our next topic for next week. Great. All right. Share and share. There we go. Okay, that's Bruce's slide, introduction. See if I can get to the next one here. Okay, session 2 is next week, October 17th. It will we'll have the room open starting probably 11:30 or so. So if you want to get your connection set up and join us, anytime between 11:30 and noon is fine. The reminder, I know that went out said 11:30, but that was just to alert you that the space was open and you could join, set up your stuff, step out, get your lunch, cup of coffee, whatever you need to do, and then we're game on at noon. The topic for next week's session is novel trait development. I'll be hosting again. We'll have 2 speakers, kind of back to our traditional structure here. The first half will be a presentation by Dr. Scott Spidell at Colorado State, and he's going to discuss the genetic evaluation for PAP, or pulmonary arterial pressure, so high altitude disease, and the work they've been doing there at CSU. And the second part, my colleague here at K-State, Dr. Megan Rolfe, will discuss water intake. She's got a USDA grant looking at individual animal water intakes and adaptability, and so looking at testing period and within animal and across animal variation in water intake. So I think 2 really interesting topics, but we'll have those for next week's session. In the meantime, if you, I think, should have an email in your inbox with the full program schedule. Looking forward to weeks 3 and 4 as well. But we look forward to seeing you next Wednesday at noon Central, October 17th, and we'll close for now. Thanks everybody for joining us. Bruce, thanks much from our team and the participants for your comments today. We really appreciate your engagement.
Yeah, thank you.